welcome to Good Eats. Today I'll be showing you how to co-transcriptionally splice pre-mRNA into mature mRNA. So let's get started. So to begin, we should probably start off with a little bit of some DNA. Then let's add a few transcription factors. Bam! Bam! Now also some splicing factors. And also, you can't forget your RNA poly 2 along with CTD. In addition, we're adding some capping enzymes and some polyadenylation factors. Now we're ready to cook. Now, you just pop in the oven for like 20 to 30 minutes and you'll have your nicely cooked mRNA. But let's see what happens, what's going on in the oven right now. To begin, TBP, which is also known as Tata box binding protein of TF2D, binds to the Tata box. Then TF2A soon follows and helps with stabilizing the TBP on the DNA. Then TF2B comes in and is the RNA polymerase II recruiter and marks where transcription will start. Soon after, RNA polymerase II binds to the DNA along with its companion CTD. Following RNA polymerase II addition, TH2F, TH2E, and TF2H complete the PIC assembly. What happens next is that the TF2H acts as both a helicase and a kinase by first unwinding DNA and then phosphorylating the CTD. The CTD, otherwise known as the carboxyl terminal domain, is part of the RNA polymerase II complex. It plays a role in transcription regulation as well as binds factors for pre-mRNA capping, splicing, and polyadenylation. Once phosphorylated by TF2H, this signals a transition from transcription initiation to elongation. It as well recruits RNA processing enzymes that act during elongation, which include capping enzymes, splicing factors, and polyadenylation factors. Now we're ready for elongation, in which TF2B, TF2E, and TF2H leave the complex. As elongation begins, pre-mRNA meets the capping enzyme from the CTD, in which the 5' end is methylated, creating a 5' cap. As elongation continues, splicing of the intron and exons of the mRNA initiates. But let's take a closer look. Key points or locations are the 5' splice site of the 5' exon, the adenine branch point site of the intron, and the 3' site of the 3' exon. This splicing is done via transesterification by a protein complex called the spliceosome. The spliceosome is composed of the 5 snRNAs that were added earlier and are recruited to the pre-mRNA template by CTD. The first spliceosome component is U1, recognizing the 5' cap splice site. U2 then binds to the adenine branch site located on the intron via ATP hydrolysis. Then the U4, U5, U6 complex joins. U6 then unbinds U4 and base pairs with U2. And as well binds to the 5' splice site, displacing U1. Then once the splice sites are aligned, the first transesterification can happen. The free 2' OH of the branch site does a nucleophilic attack on the 5' phosphate of the 5' exon, in which it releases the 3' end of the 5' exon. This forms the laureate loop structure. Then the second transesterification is carried out with the free 3' hydroxy group of the 5' intron, in which it attacks the 5' phosphate of the 3' exon. Then the intron laureate loop structure is released, forming a new mature mRNA. Now, as a side note, it's important to keep in mind that sometimes not all the introns are spliced out, and that sometimes not all the exons are bound together. Sometimes there's alternative exon splicing. This can be a result of a variety of things, such as protein binding affinity to the splice sites. There's a splice site here, 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 and here. An example is the sex-lethal gene of the Drosophila. What happens here is the SXL protein binds to one of the 4 splice sites. 1, 2, 3, 4. In this case, the SXL protein binds to the second splice site. Now, if the SXL protein was not present at that splice site, all splice sites would be cut and the middle exon would appear in the mature mRNA. The ultimate effect of this alternative splicing is that it will decide whether Drosophila will express male or female proteins. Female with the expression of only the 1st and 3rd exon, and male if it expresses all exons. The main point is that alternative splicing can lead to a variety of gene expression and function. Also, some mistakes can happen when trying to prepare mRNA. At times, you could have unwanted introns unspliced within the mRNA. How might this happen, you might ask? Well, say you have a mutation in the intron that you're trying to splice out, and that a mutation would cause there to be a new splice site within the intron, say in the middle of this intron, for instance. This might allow the intron to be included into the mature mRNA. By including this unwanted intron into the mature mRNA, this can lead to serious problems, such as blood disorders that deals with hemoglobin, and which would have a detrimental effect on the human body. Another thing to keep in mind about this whole co-transcriptional splicing process is that the rate at which the mRNA is being transcribed will ultimately affect the rate at which splicing will occur. So if transcription were to slow down, the rate of splicing would consequently slow down as well. This is seen when a nucleosome binds to the single-stranded DNA, and this causes the polymerase to slow down. Now getting back to the bigger picture, elongation continues until the RNA polymerase recognizes the cleavage site, which is recognized by AAUUAA, which is then cleaved by an endonuclease. Poly A polymerase is recruited to the poly A site and carries out the addition of adenines to the 3' end of the mRNA, creating the poly A tail, completing the transcription process. Now, once you've done all that, you're ready to take it out the oven. Now, you got a delicious string of mature mRNA that you can show all your friends at home what a good job you did. Tune in next time with our special chefs Jacob Rivera and Kai Kong, in which they'll turn this mRNA into protein.